As we head into our last session, we are changing gears and introducing our implementation team. Again, I'm happy to see that so many people are joining us for our last session of the day. Again, the focus for this event is really the intersection of research and community, and how participant data is gathered and used in research, and how that data is being used to improve patient care, which is our last session today. So I'm very happy to be joined by 3 members of Precision Health Implementation Work Group. This is a group that takes findings from the research being done at the University of Michigan and finds a way to work through all the processes to make changes to clinical care. It's the final step in a long translational process from data collection from patients, analysis, and research to return actionable change into the healthcare system. And so joining me today is Jessica Verzi, who's a clinical, informaticist working in Precision Health. Amy Pasternak, who's a clinical assistant professor in the College of Pharmacy and a clinical pharmacist. And Erin Odles, who's a medical scientist training program fellow. Again, similar to all of our other panels, if you would please put your questions in the Q&A. After our panelists are done speaking, we will then answer your questions. And with that, Jessica, I leave the screen to you.
Thank you. Thank you everyone for joining. Can you hear me okay?
Yes.
Awesome. All right. So before I begin, I did want to extend a very warm welcome to the participant community for joining us today in this discussion. I'm excited that you are here, and I'm very delighted to be here as well. My name is Jessica Verzi. I am a member of the Precision Health Implementation Team. Today, my discussion objectives are to provide an overview of how the Precision Health Implementation Team supports the translation of precision health discoveries into clinical practice at Michigan Medicine. I will also introduce 2 fabulous colleagues of mine who will provide an overview of real-world examples of how precision health discoveries are occurring at the point of care at Michigan Medicine. In addition to that, we'll spend a little bit of time discussing how precision health embraces diversity, how your unique data contributes to precision health discoveries. So we'll kick this off by talking a little bit about precision health. Throughout the day, you have heard awesome presentations about the discoveries, and they seem to be boundless and very exciting. So precision health is an emerging approach to individualized healthcare delivery. Precision health discoveries can better predict, prevent, treat, and manage disease that is unique to you and your family. Precision Health empowers researchers and your medical team to develop unique prevention and treatment strategies based on the following factors that are unique to you. As a Precision Health community member, you may have participated in a research study where you've donated a blood sample for DNA analysis. You may also have completed questionnaires that detail your family health history or your medical history. In addition to that, another great example of collection of this very important data is if you visit your primary care physician, and oftentimes during those visits they collect family health history and medical history as well. Those data points are stored in your electronic healthcare record and easily extractable to the researchers and medical care teams to inform precision health discoveries. In addition to those factors, environmental factors such as where you live and your lifestyle, what your exercise patterns are or your eating habits are all contributing factors to precision health discoveries. So precision health, as you've learned today, certainly embraces diversity. Looking outside of that traditional box and developing medical interventions and treatments that are customized to meet your unique personalized health and wellness plans. So going beyond that one-size-fits-all approach. Here we'll recap a little bit about how your unique data contributes to precision health discoveries. Again, at the heart of precision health, prevention strategies. So your data helps determine the best individualized interventions and treatment plans that are best for you based on your background. In addition to that, your data helps determine which treatment may work best for you. Patients with different backgrounds and genetic backgrounds, there's certainly a science to that that Amy will talk about in a little bit more detail later on in the presentation. Your data also contributes to prediction strategies. So how to identify your medical and genetic risk for development of medical complications and diseases. A great example of a medical complication that Erkin will go into in a little bit more detail later on in the presentation is maybe your risk factor for developing a hospital-acquired infection, such as C. difficile or C. diff as commonly known, or maybe a little bit more serious, maybe detecting your risk for developing a septic infection. So let's meet the team. What happens with these discoveries and how do we translate these discoveries into clinical practice at Michigan Medicine? Again, I'm Jessica Verzi. I'm a nurse by background and I have practiced my nursing at Michigan Medicine. In addition to that, I focused on many quality efforts and research, of course, that certainly impact patient care, best outcomes, et cetera. I'd also like to introduce Dr. Hemi Cho. Hemi Cho is a Associate Director of the Implementation Work Group. She is also the Associate Dean and Chief Quality Officer for the University of Michigan College of Pharmacy and the University of Michigan Medical Group. Dr. Paul Grant, another great colleague. He's the assistant director of our work group. He is also a hospitalist in the Division of Hospital Medicine at the University of Michigan. In addition to, he is an associate professor of medicine at the University of Michigan Medical School. But we can't do this alone. So in the next slide, we'll talk more about what we do and how we do it. The overall goal of the implementation work group within Precision Health is to develop a process for integrating or implementing, translating these precision health discoveries into practice at Michigan Medicine. The precision health participant community, Michigan Medicine researchers, medical care teams, and leadership are key to successful translation of these discoveries. It certainly takes a village. Over to the right, I just want to walk you through, the process, a high-level process of what it takes to take this discovery and translate it into clinical practice. And it all begins with that precision health discovery. So perhaps the discovery is related to prevention and prediction, or perhaps the discovery is related to developing the best treatment options for a particular patient population. Once we determine what that discovery is, we begin to work with Michigan Medicine researchers and the medical care teams at Michigan Medicine um, to demonstrate the impact to patient outcomes. So we do some analysis on that, the richness of the data that we talked about, the contributing factors to precision health. We do some data analysis to determine how well does this discovery predict, or how well does this discovery determine the best treatment option for a particular patient population. We then move into disseminating the findings, and this is where the precision health team really, is that liaison and that bridge to bringing the researchers and the Michigan medical care teams together to discuss the best approach to bringing this discovery to the point of care. And we do move on to clinical implementation. And this is where we actually translate this discovery into practice. A great example of this may be from a prediction perspective, maybe we've developed a tool to help medical decision-making that your Michigan Medicine provider would use to inform healthcare decisions. Maybe, like I said, maybe detecting your level of risk for developing a hospital-acquired infection, or detecting your level of risk for developing sepsis. There's always a continuous improvement loop in this process, always. This is very important, an important process step, because this allows us to again, analyze to make sure that the discovery is demonstrating positive patient outcomes, demonstrating that positive patient impact. At this point, I'd like to turn it over to my colleagues, and you'll learn a little bit more about those real world examples that I spoke of earlier. Amy will talk about those precision health discoveries as they're related to treatment, and Erkin will talk about the precision health discoveries that are related to prediction and prevention.
All right, thank you, Jessica. Can everyone hear me okay and see my slides?
Yep.
Okay, so good afternoon, everyone. My name is Amy Pasternak. I am a clinical pharmacist at Michigan Medicine. I'm very excited to be here with you this afternoon to share some of the work I've been doing in collaboration with the Precision Health Implementation Working Group in terms of integrating pharmacogenetics and actually implementing this information into clinical care here at Michigan Medicine. To get started this afternoon, I wanted to quickly take a survey just to get a sense of how familiar you all may be with pharmacogenomics. So if you could please just take a moment to respond to the poll by selecting the answer that best fits your current knowledge of this area. Okay. So it looks like most have at least heard of pharmacogenomics, but are probably not that familiar with others who are not very familiar with what pharmacogenomics is. And so that's great. That's the goal of this afternoon, is to teach you more about what we're doing and what we can implement with this information. So my goal today is really to describe pharmacogenomics, particularly for those who may not be as familiar with what this field is, and then take a little time to describe how this topic relates to the implementation of precision health, specifically by describing some examples of how we're beginning to use pharmacogenomics as a tool for precision health in the Michigan Medicine record, and also describe some future opportunities of using the MGI data to improve our pharmacogenomics integration with precision health overall. So for those who are not familiar, pharmacogenomics is the study of how genetic variation impacts a medication response. And so this could evaluate both why patients have positive responses to medications, as well as why patients potentially have unintended responses, such as developing side effects to medications. And so although performing research to better understand why patients have different responses is important, I think the key here, particularly as we're talking about implementation, is what does this actually mean in terms of translating this into patient care? And so by using the research resources we have available to increase our understanding of how genetics impacts medication responses, we can then take that information and begin integrating that genetics into our precision health strategies to better determine potentially the best drug or the best dose of a drug for a patient, so that their medication therapy is personalized to their genetics based on their need. And so pharmacogenomics is just one example of the overall precision health puzzle, making sure that the patient's receiving the best treatment at the best point in time. So I'm a very visual learner. I kind of like to think through these things with visualization. So, think about a time when you needed to take a medication. You probably bought an over-the-counter product, read the bottle instructions, and took a dose of a medication based on what the bottle recommended you do. And so for this example, let's think about drug X. And drug X is used to help treat headaches. And so all of our patients on this screen currently have a headache and need to treat it with this medication. And so based on the bottle description, all of these patients would take the same dose of this drug. And although they all have the same problem and they take the same dose of this medication to treat it, what we ultimately see frequently is that not all of these patients will have the same response to the medication. And so for most patients, fortunately, the drug works really well and their headache goes away and they have no further problems. But for some patients, we find that this drug really doesn't work at all and potentially their headache persists and they're not getting any relief. Or for other patients, the drug may have some unintended consequences, such as developing a side effect, potentially nausea or vomiting as an example, in terms of taking this medication and it not doing what we expect it to do. And frequently when we're thinking about choosing a drug for a patient, we don't always know ahead of time which category or which response the patient's going to have. So oftentimes it's that one size fits all approach that Jessica mentioned in her previous slides, in terms of everyone gets the same drug to try to treat the same condition. However, as we start to learn more about pharmacogenetics and can apply more pharmacogenetics, we can start to understand who may respond a certain way to a medication prior to prescribing that drug to the patient. And so by knowing your genetic information ahead of time, we're able to potentially determine if you may be at a higher risk for side effects, or potentially if you're more likely to respond to a certain medication. And the goal is that we can then integrate this into your care to be able to tailor any prescription decisions to be personalized to you to improve the chances that your medication will work well. So I wanted to go through a couple of basic examples of how we're beginning to integrate the available clinical pharmacogenetic information we have here at Michigan Medicine. The first step of this process is we're really leveraging the electronic medical record that we currently have. And the first thing we might consider is identifying patients who would benefit from having a pharmacogenetic test based on their prescription history. And so we can use the electronic medical record to identify patients who may benefit from testing based on some characteristic that we can identify, such as the placement of a prescription for a certain medication. When the medical record identifies that this prescription is being ordered, and we have alerted the medical record that this prescription should have an associated pharmacogenomic test, we can then deploy a recommendation directly to your provider at the time of the prescription to make sure that that test is ordered for an assessment prior to prescribing. In addition to identifying whether a pharmacogenetic test may be needed, once the pharmacogenetic test result is available in the medical record, we can then further identify which patients may benefit from a medication adjustment, potentially receiving a different medication or a medication dose adjustment, to avoid something like a side effect. So in this example, let's say we have two patients who we identify carry a genetic variant for the pharmacogenomic test. In the medical record, again, we can link that pharmacogenomic result and the medication order together to notify your clinician that there are personalized recommendations for your therapy at the time that the prescription is written. So like I mentioned, we're currently developing these processes to further integrate all of the clinical pharmacogenomic information that's available within our medical record. As researchers, we've also identified that there are many important pharmacogenomic markers that we can study within the Michigan Genomics Initiative. And it has been mentioned in prior presentations, our goal is ultimately that we would like to translate as much of this information as possible into clinical practice to improve patient care. But there are some limitations in terms of using the MGI data to inform actual prescribing practices in terms of pharmacogenomics. And so there are multiple researchers currently working to develop studies where we're able to use MGI data to help identify whether a patient may be a good candidate for a pharmacogenomic research study. And I just wanted to quickly walk through one example where this has been activated in a clinical research study. So we recently opened a study called the Confirmatory DPYD Testing in MGI Subjects. This study is being led by my colleague, Dr. Daniel Hertz at the College of Pharmacy. And the purpose of this study is to help identify patients who may be at risk for increased side effects with certain medications. The gene being focused on in this study can help predict the incidence of severe side effects to certain types of chemotherapy. And so this study process was developed to try to utilize MGI to identify patients who may be at increased risk for side effects, then identify whether or not they're actually likely to receive that medication. And if they are, these patients will be invited to participate in this research study to obtain the confirmatory clinical testing that would be needed to apply this information in their daily care practices. And so we're very excited to have opened this study and see the results of this, and are looking forward to future studies where we can use the MGI data to identify patients who may benefit from pharmacogenomic testing. Thanks very much for your attention. At this time, I'll turn over to our next speaker and be happy to take any questions during the Q&A session.
Good afternoon, everyone. I got my presentation going. Can everyone see that okay and hear me?
Yes, both.
Excellent.
Yay, thumbs up.
All right. So thank you guys for joining us this afternoon. I'm very excited to talk to you guys a little bit about how we power precision health through prediction and prevention. So my name is Eric Notless. I am a MD PhD student. I am co-advised by Dr. Jenna Wiens in computer science and Dr. Brian Denton in industrial and operations engineering. And today we'll be talking a bunch of stuff, but at the tail end, we'll highlight some work that was done in collaboration with some of my colleagues in the MLD3 lab, as well as Ben Lee and Dr. Krishna Rao, who are in the medical school. So when I think of precision health, I think of this diagram. And this diagram is sort of a simple way of how I put together the different parts that we need to have a successful implementation for a model or for a technique that we'll be using that's been developed through our precision health approach. And so the three components that we have here are the patient, the physician, and a prediction. And the patient and physician are probably familiar to everyone in the audience. In fact, they're sort of the core foundation of what we call shared decision-making, or our approach of how we make good decisions for our patients. Well, we have both the patient and the physician come together with their shared sets of knowledge, and they come up with the best decision for the ideal outcomes for the patient. Now, I have physician there, and it really isn't just the physician, it's the whole clinical team. It's everyone that might be helping to deliver care, be that the doctors, nurses, respiratory therapists, occupational therapists, anyone that's on the clinical team. I used physician just because I like the alliteration of the diagram. So the third part is the prediction, and that's what we're going to be spending most of our time for the initial part of this talk talking about. The prediction is sort of key to precision health. So what is prediction? Well, everyone probably interacts with predictions thousands of times every day. You just might not, see it sort of up front and laid out in front of you as a prediction. But when you open up your Netflix screen on your TV or an iPad, what's going on is Netflix behind the scenes is making, tens, if not hundreds of different predictions where they are trying to figure out what content to serve to you based on what you've seen in the past so that they can get you to engage with their content and then pay them in perpetuity to continue watching their content. And then when I was preparing for this talk, I used Google, and Google is notorious for their predictions, right? They're using predictive analytics at various stages in their products, in their app, and even in their search, right? That's core to their search. But even when you're thinking about what to search, right, they take what you're thinking about and then they are serving you back things that you might be interested in searching. So that search initially was displaying things like preeclampsia, prednisone, things that me as a medical student might be interested in searching. And then they're also, you know, giving me some things about food and working out. These are all my hobbies. And because they know so much about me, they're able to give me relatively good starting points for queries. So what is prediction? We have a sense of its pervasiveness, but fundamentally at its core, it's based on the concept of, if we know some stuff right now and we have some information available to us, is there a way to figure out something that we don't have available to us? Is there any way to recover information that we may not know directly at this point in time? And so how do we make predictions? Well, if you had a crystal ball, you could use that. Unfortunately, I'm not so lucky, and my other ESP perceptions are relatively limited, so I use data-driven models. And so what is a model? Well, when we build models for precision health and when we deploy them, we think about them in three big components. There is the input. This is what we know. And these can be things that exist in the electronic health record that might be being collected routinely all the time. So things like age, heart rate, comorbidities, medications. We then have a model. We pass this information either from the electronic health record or other systems into a model that we've built. This model could be just a bunch of rules. It could be a machine learning model that we've trained. It could be an AI algorithm. There's lots of different names for models, but you can think of them as a black box that you passed your input to. And then once you pass your input to the model, it'll evaluate and give back an output. And this can be a lot of different things, but in precision health, what we're really looking for is often a prediction. And what kind of predictions are we looking for? Well, things that might be important for medical decision making and important for physicians and patients to know. So one of the big ones could be potentially the risk of a patient developing diabetes in the next 5 to 10 years. It could be the length of stay of a patient that is about to be admitted to the hospital. All these would be potentially unknown information, but would be useful for either a physician or patient. And so now we have a concept of how the models work and their sort of components, but then how do we go about building models? This is a key part of what we do in terms of our research and then what we translate into our work at Precision Health. And that is you either have a lot of data or you have a lot of knowledge. And in medicine, in healthcare in general, we have a lot of both. We have data from the electronic health record, and we have highly trained nurses, physicians, and allied health professionals that have a wealth of clinical knowledge as well as experience that they can sort of use to guide decision-making and prediction-making abilities. So what you can do with these two approaches, they're complementary, but they sort of yield different types of models. So on the left, what I've done is I had a bunch of data from a bunch of athletes where I knew their height and I knew their weight. And then let's say I wanted to figure out a way to estimate the weight of a new athlete that I've never seen before in a database that I'm building, or I'm interested in figuring out some information that I don't know about them. Well, I had a bunch of data and I was able to build a best fit line that essentially just maps the unknown, which is the weight, to the known, the height. And this may be a throwback to physics class or statistics class, but really very simple, just like linear regression or best fit line. We do this sort of at scale at our lab with lots and lots of data from the electronic health record. And we take this and we bring it to physicians. Physicians operate sort of in a different world where they're sort of doing decisions based on theories or rules, and that's sort of represented on the right, where you may have a whole bunch of rules that you sort of follow, like an if-then series of statements. So you can see that if the patient is below a certain weight, then you'd expect their height to be a certain number. And so you can see that there's two different approaches, and you can build different types of models, and they're actually complementary to one another. So what we seek to do is to have data-driven models that when we bring them to the physicians, they help to bring sort of knowledge or insight to gaps that might be difficult from a physician's individual experience to be able to make good decisions. So we talked a lot about predictions, and now really the big focus is not just the prediction, but it's the whole system. It's the relationship between the prediction, the patient, and the physician. And so the real question is not how do we make predictions, right? We can go into a long talk about the math. My advisor told me absolutely not, I cannot talk to you about math. So unfortunately, we're going to talk about utility, which is actually a lot more important. So you can think about many different ways to build different machine learning models that sort of fit this intersection, the center point of the Venn diagram. And there's things like building risk scores, there's building prognostic models that might tell you how long you expect a certain patient to experience a certain disease. You could help physicians with treatment selection. You could also build screening tools. The primary thing that we're going to talk about are early warning systems. So an early warning system is essentially a system that is designed to build a prediction and serve it to physicians so that we can help prevent bad outcomes. And so, you might be thinking about, well, what kind of bad outcomes are there? Well, there's many. And in the hospital setting, there's a lot, right? So you could think about a patient that's hospitalized. You could ask yourself, well, will this patient become septic? Will they need to be put on a ventilator? Are they going to experience any risk of having a hospital-acquired infection? You can understand that probably if you have a notion of if they're at risk of any of these things, could actually probably intervene and you could prevent a bad outcome. And the sooner you intervene, the more likely you are probably of preventing a bad outcome from happening, right? So there's a lot of incentive riding on making high quality predictions for the patients that are hospitalized. And now you might ask me, well, Erica, don't physicians or nurses or anybody in the hospital, don't they think about this? And of course, yes, they do. And if you brought this list of things that you're interested in monitoring for and catching early, brought them to a clinician, they'd probably be able to do a really good job of assessing a patient if they're at risk for any of these things. But the difficulty happens when we try to do this at a larger scale, right? We're not interested in one patient at one point in time. We're interested in doing this for many patients over their whole duration of hospitalization, potentially, right? So this becomes very overwhelming to physicians who are primarily trying to treat the patients in the hospital, right? Trying to address the underlying cause for why they're in the hospital. And then you might be asking them to keep track of a laundry list of additional things for every single one of their patients. So this would be very difficult to do. But fortunately for us, we have computers and we have models that we built that can assess all the patients that we have in a hospital or in a unit, and then give an assessment of their risk for each of the questions. And then they physician or nurse or anyone else, and then have them intervene to help prevent the bad outcome. So that was sort of abstractly what we do. More concretely, I'd like to give you guys an example of one of the early warning systems that we've been working on. So this is for hospital-associated C. difficile infections, also known as CDI. And CDIs, especially ones generated from the hospital, are a big concern because the ones that come from the hospital could potentially have been avoided. The ones that there's another class that come from the community, and we don't have a lot of control inside the hospital to do anything about those. But the ones that happen in the hospital, we really like to prevent because there are 200,000 cases per year in the United States of hospital-acquired C. difficile infections. And of those 200,000 cases per year, we have 16,000 deaths. And so this is a relatively big burden. And even for the patients that don't experience the worst outcome, the outcomes sort of on the lower end of the scale are still significant, ranging from diarrhea to colectomies, a surgery where you remove parts of the colon. So this is a big deal and something that we'd like to prevent. So what we've done is we built a series of models at a couple different institutions, one here at University of Michigan, another at Mass General Hospital, where we took 250,000 historical patient records and we built a model that uses both the patient info and the unit info and passes that into the core model. And then that model gives us back an assessment of the CDI risk of a given patient. And it's built such that we can pass information for every patient for every day. So it's able to keep track of how patients are trending, if they're trending towards potentially being infected by this dangerous bug. So schematically, what's going on here is you have the patient and the physician, and you have many patients and potentially many physicians in the hospital. And every day, information is coming from the electronic health record. It's getting passed to our model. Our model then uses things it knows, things that we've trained it on, where it says, you know, if you're younger, you're probably lower risk. If you're older, you might be higher risk. If you've had CDI past, you may also have higher risk. It takes all these things into account, and then every day it generates a risk score for each patient. And you can watch this risk score every day. We have a threshold where if after a certain point, if the number gets above this threshold, then we would sort of consider intervening. You could think about a whole bunch of different systems, right? You could send a page to the physician once you hit this threshold. The physicians could potentially look at a list of all the patients and look at their scores and say, I'm worried about this patient. They might be at risk of CDI. And if they're at risk of CDI, the patients could have their treatments adjusted by the physician. Or in order to protect other patients, they could take a patient that is suspected of being infected and isolate them from other patients so that those patients in the hospital are not at risk of getting it from the suspected individual. So there's a lot of different things that could be used with that information, right? So it's all about delivering crucial information to the physician and patient that is actionable and can prevent a bad outcome. And so we have in our lab and in our work with the Precision Health team, we have a lot of different projects going. One is the C. difficile early warning system. We have another similar early warning system that's actually built for patients that come into the hospital with COVID-19, and we're interested in seeing who may potentially, decompensate, have a bad outcome. But there's also the flip side where we can patients that might have, might not need that much support and may actually be relatively safe to send them home early because this is an unknown disease. We don't necessarily know what the trajectory looks like, and the model is able to help us figure out who we can safely send home early and clear up room in the hospital and help keep those patients safe from hospital-acquired infections. And we're also working on early warning systems for sepsis. And then our work is not limited to just chronic care. We have some projects on predicting who may be at risk of developing Alzheimer's disease, who's currently presenting with mild cognitive impairment. We have projects on diabetes management, and then also helping to predict who may recover from occupational injuries. So with that, I'd like to just leave you with the Venn diagram, and really remember that what we're trying to do here is build predictions that serve both the patient and physician. And I believe now is our time to, one, thank you guys for listening to me talk, but also start to open up the Q&A and hear what your questions are so that we can answer them and hopefully share some knowledge. Thank you.
Great, thank you so much for that. That was an absolutely wonderful presentation. I think I'm going to have all the panelists come back now. Again, if you have questions for our panel, please use the Q&A tab. Otherwise, I will start off the questions. We have one right now. So how do you quantify the potential clinical impact on patient outcomes? How do you decide a new risk predictor, whether it be a genetic test or other test, is worth giving back to the patient? Because we clearly don't want to go after everything, because there's a lot of science out there that we find at one point we think is really important, but it ends up not really being that important.
I'll kick it off. Can you hear me okay?
Mm-hmm. Yes.
All right. So some of what we decide from a patient impact perspective and, promoting best outcomes, some of that comes to us in ways where, we're aligning our priorities with the health system priorities. So if the health system, the medical providers at the health system are interested in improving sepsis, for example, or detecting risk of sepsis, we would certainly look at it from that perspective as well. I mean, So there's a quality perspective, but there's also a patient safety perspective that really is number one. So, if there is something that is brought forward, we would certainly prioritize patient safety first.
Thank you, Jessica. And I think to kind of follow that up, I think that that's really, the patient safety is certainly something that moves things forward very, very quickly. And Amy knows the details of this better than I do, but I know that the DPYD project that's going on in terms of the number of patients that this potentially will impact is probably fairly low. But the severity of what could happen if it's not known is fairly high. Do you want to talk a little bit about that?
Yeah, so I was actually thinking the same thing in terms of echoing. When we think about in terms of selecting right now which things we're going to return, we're really focusing on those with really strongly associated safety concerns, because we know that there can be severe consequences if we don't try to address that. And so when I was talking about the items that we have worked on integrating into the medical record on the clinical side, again, we're focused on those where there is a safety concern, where we can prevent a serious adverse event from occurring by identifying this information in advance of prescribing the drug. And hopefully, like Vicki mentioned, with this newly open study, being able to utilize that information to further identify patients at risk for severe adverse events.
Erkin, do you have any thoughts about how moving things into the clinic, kind of the threshold for doing that?
Yeah, so I think, echoing what Jessica and Amy said about safety being sort of tantamount, I think that's like the minimum bar that all models and anything that we're thinking about implementing, everything has to meet that. And for some of the stuff that we've been working on, we actually do a lot of study in a lot of different steps before the model will even be considered to be something that will pass off to a patient or a physician. And I didn't really talk about it in our presentation, but maybe I'll spend like 30 seconds on it right here. So when we build a model, we assess the model and see how well it works in terms of our intended target performance, right? So we want it to perform well. And then what we'll do is we'll often look and see, how does it work in certain populations? Is it not very good at working on men that are aged 25 to 35 and are white, right? Or is it targeting certain graphics that might not be working well in? So we have all kinds of tests that we'll do before the model even really leaves the workshop, so to speak. And then there's a couple more steps before the model even will get to deployment. And one of them is like, we'll have the model running in the background and watch how well it works without any information being passed to a physician or patient. And so that's where the CDI model is right now, actually. It's been sort of deployed in shadow mode for probably over a year now. And we're doing a lot of work to assess, to see how well the model is working, because we want to be absolutely sure that the models that we put out don't cause any harm, or to understand, what the blind spots might be on those models. And then the next step will be, once we're happy with where they're at, then we figure out how do we plug it in and how do we make sure that physicians and patients know, how this model is supposed to be used and what to watch out for when they're using it.
That's a really great way of looking at it, kind of doing the silent models for a while to see what the overall impact is. I think I'm going to throw this back to Amy again, because one of the things that has really been at the forefront of the implementation is the pharmacogenetic testing that goes on throughout the country, and the fact that this is information that the entire country is interested in, and there's many direct-to-patient or direct-to-consumer testing companies out there and ways to get this information. And I think one of the things that is really, had Michigan put a pause on that was wanting to make sure that the evidence behind that was really solid, because there were costs associated with the tests as well. And so not wanting to unnecessarily spend healthcare dollars nor spend personal dollars through copays. So Amy, do you want to talk a little bit about some of the broad pharmacogenomic testing beyond DPYD?
Yeah, so when we were looking at our clinical implementation, I think Erkin brings up a great point of, the validation is a huge component of this. We can't really have one study that says there may be an association between a gene and a drug response. We really need lots of data to prove that this is a true response we're seeing in different populations. So that was one component, and we're very fortunate in the pharmacogenomics side in that there are some existing resources that we can use. The FDA, which is responsible for approving drugs, does provide some guidance in terms of certain medications that should be evaluated for a genetic component prior to prescribing, and so we're able to leverage that to show that association. There are some groups that provide guidelines in terms of really showing there's strong evidence between a gene and a drug. But the really important thing is when you go to make a recommendation, you have to know what you're going to recommend. Are you going to recommend using an alternative drug, or are you going to recommend adjusting a dose for a drug? And so there's lots of time spent evaluating that, discussing that with the clinical groups in terms of how does this work into your practice, how will this impact your patients, and then assessing the cost of the testing as well, and trying to work to figure out the most cost-effective method to improve the availability of this information to make sure it's safe for patients.
I think, along the lines of that as well, it goes back to what Chad Brummett discussed earlier, looking at going back into MGI and trying to understand some of the genetics behind people that respond or develop the phenotype of misusing opioids. And I think then that too kind of circles back into how, Michigan Medicine is trying to use the tremendous gift of data that all the research participants have given us to actually impact patient care, and that's not an easy it's not an easy question to answer, partly because pain is so subjective. But I think with this resource, we'll be able to begin starting to have some of those answers, and putting these processes into place will have effective ways to implement it once that data reaches the thresholds where we can put it into clinical care.
Yeah. I just want to echo that really ties into what Jessica said in her presentation in terms of the continuous improvement process of this. We are just starting to deploy these types of interventions, and we will constantly be assessing what other, in my perspective from pharmacogenomics, what other medications can we make recommendations on, or how can we fine tune these recommendations overall.
Well, and I think that's the beauty of the scientific process and the scientific method, in that we're continually getting new data and coming to additional conclusions. And sometimes it's very frustrating, but it's part of the process, and it's a continual improvement process. I think is a really great way, all striving for better health outcomes and better patient safety as well. I'm not seeing any more questions in the Q&A, so I'm gonna give a last call for questions before I thank our panelists. Are there anything anyone in our panel would like to say before we switch to our last speaker? Okay, not seeing any questions. I want to thank you all for participating in this panel. It was an honor today to be your moderator. And with that, I want to also thank everyone for attending. We have one last speaker. And so I'd like to introduce Dr. Jenna Wiens, who's going to help us wrap things up. She is an associate professor of computer science and engineering in the College of Engineering here at the University of Michigan. And she's also a co-director of Precision Health. Jenna.
Thank you, Vicki. So that concludes our virtual Precision Health Symposium. And on behalf of the entire Precision Health team, I want to thank all of you for participating in today's event. We'd especially like to thank all of our speakers, panelists, poster presenters. If you missed anything, the meeting was recorded and will be made available shortly. In addition, the poster site will be live for the next month. And while the presenters won't be there the entire time, you can certainly return to their posters and contact them with any follow-up questions. A lot of effort went into making this event look effortless. And for that, I'd like to thank Vicki Ellengrad and Tina Krager and our team at Precision Health. They definitely deserve a round of at least virtual applause. So thank you. And last but not least, I'd like to thank all of the research participants. Without your willingness to participate and engage with Precision Health, many of the research advances that you heard about today would not be possible. And I hope to see you all at next year's Precision Health Symposium. Thank you. Bye, everyone.